excited about that. Literally from all over North America and the globe. Okay, my clock says it is straight up 12 noon, and I'd like to welcome everybody to the I-29 Moo University Dairy Webinar Series. Today's presenter is Dr. Kaitlyn. She's an animal science researcher with USDA ARS National Animal Disease Center here in Ames, Iowa. And she'll be discussing the results of several studies to define the kinetics of protective immunity in cattle that have been infected with avian influenza. As I always say, the podium is yours, Dr. Kaitlyn.
Thank you, Dr. Fred. Thank you for the opportunity to be here today, and thank you to everybody who's tuning in. So I'm just going to give a quick overview of what it is that I'm going to talk about. So I'm going to start with just kind of an overview of influenza, because it kind of gets to be where it's its own language. And so making sure that we are kind of all on the same page about terminology. Then I'll talk about our studies we've done and what we've learned about lactating cows, what we've learned about calves from our studies, and kind of what's next. And then I'll leave time for questions and feedback as to what you would like to see come next. So I'm going to start with just some influenza virus basics, because if you're like me, you were maybe not super familiar with all of these nuances before this became an issue in dairy cattle. So there's 4 main types of influenza. They are very creatively named A, B, C, and D. You're probably most familiar with A and B. Those are your human seasonal influenzas. So if you're getting your flu shot, that's typically what's in those. A is the only one that's been known to cause flu pandemics. What's interesting is influenza D is the one that primarily affects cattle. It can sometimes spill over into other animals. It doesn't really infect people, at least in a way that causes illness. And until this outbreak, we really thought influenza D was the only type that it infected cattle. And so I'll talk about kind of just how surprising it was to find this virus in cattle and kind of how that came about. So some basics kind of about how we name influenza viruses. So this is within those influenza A's. They are divided into subtypes based on their surface proteins. So there's the hemagglutinin or H surface protein that's shown in this picture in blue. And then also the neuraminidase, or the N, that's shown here in red. There's actually 18 different H subtypes and 11 different N subtypes. You can imagine with different combinations, you get a whole lot of options. There's been over 130 subtype combinations that have been identified in nature. And the other thing that makes this difficult is that influenza viruses can do something called reassort. So if 2 viruses infect the same animal or individual, they can actually exchange pieces of their genetic information. So influenza viruses are really kind of nefarious in their ability to change and infect quickly. So what is HPAI, or highly pathogenic avian influenza? So it is an influenza A. It's specifically an H5N1. So those are those surface proteins. And why do we call it highly pathogenic? Because it's a strain that's deadly to domestic poultry. So as I get into our cattle work, that's not what we've seen in cattle, but because it was named for obviously being found in avians and being highly pathogenic to them, that's why we refer to it as this. Strains of HPAI have been detected in the US since 2021. And before that, it was widespread across Asia and Europe. Different strains of this HPAI H5N1 have been detected in the US in mortality events in wild bird species and some wild mammals. And so just to kind of really zone in on how these different things are named, because it can get overwhelming and confusing. So within the influenza A, we've got those subtypes we just talked about. So we know this is an influenza A H5N1. Then it's going to be broken into clades and subclades. So you get into these really difficult to remember number sequences. And so what I think about to try to keep myself straight, especially coming from a genetic background, is this basically is like the flu's pedigree. So it tells us, instead of what bull it's out of, is kind of where it's come from and where it's been found and who it's closely related to. So So HPAI was confirmed in dairy cattle in March of last year. So we're coming up on 1 year. It was first identified in Texas. And so it's specifically this H5N1 clade 2344B, genotype B313. And so all of the work that I will talk about has been with this strain B313. Since the initial outbreak, this has spread to be confirmed in almost 1,000 herds across 17 states. With all of the information today, I will try to include, like here, where we have the link to where this came from, references in case you want to look up any of this later. So I talked about how we really didn't think cattle got influenza A at all. So how did we come to diagnose this highly pathogenic avian influenza in dairy cattle? So it began last spring as a multi-state investigation into this large drop in milk production. And it was kind of characterized by discolored milk. So here you can actually see some of our experimental samples. And you can see from the 2 infected teats of this cow, you have kind of like this yellowish brown color to the milk. And we actually were giving this a numerical value based on a teeth whitening score. So you can imagine kind of the really gross yellow-brown color that this became. You also saw a drop in rumination. So a lot of farms that were using some kind of monitoring device were seeing alerts go off for health. And kind of what finally broke the case were cat fatalities. So they had fed raw waste milk to cats on dairy farms. Those cats actually became neurological and died. And one of the veterinarians on the case was like, wow, this really looks like symptoms that you would see with a cat that was infected with this highly pathogenic avian influenza. Let's test and rule this out. And that actually turned out to be what was going on here. And so because we know that it was very specifically this B313 genotype that infected cattle, some folks that I work with here at the National Mental Disease Center, this is what they do, is sequence virus. We're able to trace kind of where this virus spread based on that very specific genotype. And so this started from kind of cattle moving out of that panhandle in Texas. They were asymptomatic cattle. This is an area where a lot of replacement females are raised, and they were transported, throughout the United States. And so that's a lot of where the initial spread came from, was this movement of infected cattle. So then we were asked, okay, what can we do in an experimental setting to understand what's going on with this? So we were asked to basically replicate this under experimental conditions. I'll talk about what we found and kind of what that enables us to do. So we did an intramammary infection. So the same way I typically research bacterial mastitis, the same way we would do a bacterial mastitis challenge. I'll talk a little bit later about kind of how difficult it is to pull off a study like this. So this all has to be done in biosecurity level 3 containment. So you have very strict rules as far as personal protective equipment, what can go in and out, those kinds of things. So you can see here the cattle, and then that's actually me underneath all of that PPE or personal protective equipment. So which was funny because I sent this picture to my mom and she was like, what poor sucker had to do that? And I was like, that's me. So what we did, we infected these cows in the teat with the virus. And then we kind of followed what happened with these cows and where this virus went. And we very quickly saw a very high viral load in milk. And so what we do is a test for virus levels called qPCR, or quantitative PCR. And it's a little bit confusing to interpret those results because it's what's called a cycle threshold. So the more virus there is, the fewer cycles it takes for that machine to actually pick up the virus. So when you look at some of the data and you see these very low CT or cycle threshold values, it actually means, even though it's a low numeric number, it's a high level of virus because there's so much virus it didn't take as long to be picked up during testing. So it makes things like doing correlations and things a little bit difficult to interpret. So kind of going over just what we saw from the experimental replication very much matched what we saw in the field. So you're going to see again that discolored milk from the 2 teats that we infected that I shared earlier. They were also positive for a CMT or California mastitis test. We had cow manager sensors on these animals, so we were able to follow the rumination. So you can see here that after these animals were infected, their rumination dropped significantly. And then after about 7 days, they did begin to recover. Same thing when we looked at their milk production. So this is in kilograms per day, so kind of double that for pounds. You can see these are some pretty low producing animals to begin with, which is by design. And I'll talk about kind of what that means later. We can see they had a big drop in milk production down to one of the cows was only giving like a pound per day. And then it began to recover, but never really did recover up to what it was before. And some of that is because these were two cows that were like 300 days in lactation. But some of it is also just because of the scarring or fibrosis that occurs in the infected mammary gland. And then finally, you can see here, these are the CT values. So you can see immediately how low those values got, meaning we just had a lot of virus. And then we were still detecting virus as far as 24 days post-infection, but definitely at a lot lower levels than we were at a day or two post initial infection. We did necropsy these animals, and even at 24 days, we were still able to identify virus in the mammary gland. So these are some cut tissues under a microscope that were stained, and all these little brown guys that you see here are virus. So even after 24 days post-infection, this virus was still hanging out in the mammary gland. So it's pretty wild just how much this virus likes milk and likes the mammary gland, especially considering influenzas are typically considered to be more of a respiratory disease. And this is really the first we've ever seen a virus cause any kind of mastitis. It's typically always caused by bacteria and in some rare occasions, like in algae. We also did some more staining of the mammary gland. And what's important here is everything that's in purple is a normal tissue versus everything that's in blue is going to be fibrosis or scarring. So yes, these animals did recover, but there's some fibrosis or scarring here that's going to persist. And so they would probably be impacted and have lower milk production going into the next or subsequent lactation also. So that's the bad news. The good news is that we did see a really strong antibody response in these cows. So we were able to detect antibodies in the blood by day 7 and in the milk by day 9. And by day 14, we found that these antibodies in both blood and milk are what we would consider neutralizing antibodies. And so what that means is we take these antibodies from the cow, take them back to the lab, and expose them to the virus. And so these antibodies were actually able to bind up that virus, meaning they should really be protective. So I just kind of want to talk about what it took to pull off a study like this. So these cows were in this BSL-3 environment. For just this study, we've had several subsequent studies for 24 days. This took a massive amount of work from everybody at NADC. So we had folks on the outside mixing and bagging up TMR to bring in to feed these ladies. This building was set up to house large animals and is one of very few facilities in the world that can house large animals in these kind of security conditions. But it was not set up for lactating animals. So this is a picture that was actually featured. The AP came and did a little bit of a story about what was going on here. And I like it because you can see kind of what it took to milk this, to milk cows in this. So we were using a bucket milking system. That's why we were really looking for some lower production cows to try this in the first place. Because if you've got to milk out 100 pounds into a bucket and then weigh it, it's a bad day. Still not a great day to milk out, 40 to 50 pounds in a bucket. But still, that made it just a little bit easier to actually get this research done. And then on top of that, you can see one of my coworkers and just the level of personal protective equipment that he has to wear to meet the requirements of these conditions. And so these are massive undertakings done by a very small number of people here doing a lot of work. And there are very few facilities in North America, period, that can do this. And so what's really been impressive to me is that there is a network of these BSL-3 labs, and they communicate very well and make sure nobody is overlapping work and making sure we are answering as many questions as possible with just really limited resources to be able to do some of these things. So I'm going to transition a little bit. So the work I will talk about for most of this was done here in Ames at the National Disease Center. We're going to go up north for a minute, and I'm going to talk about some work that was done in Canada at another facility called Vito. So I talked about how we identified virus neutralizing antibodies in these cows. But we did not have the ability to continue on and reinfect these cows. And that's exactly what Vito stepped in and did. So they infected these cows, recovered them, and then at day 31 days or 1 month post that initial infection, they tried infecting them again. And so the really good news is that cows were protected against reinfection. So when they are infected and they produce antibodies, these antibodies are actually going to protect against reinfection. So I'm going to transition a little bit and also talk about what we've seen in calves and kind of how that can inform a little bit more about what's going on with the immune system. So as you can imagine, I don't think I have to tell anybody who's on Moo University that milking cows is a lot of work. We wanted to explore, can we look at non-lactating cows and can that tell us anything? So it was a big question also is that since we've seen so much virus in the milk, we saw these cat fatalities from cats consuming raw infected milk. Can milk be a route of infection in calves? So that's exactly what we did here at Ames. So we had 2 experimentally infected cows. We milked them and then bucket fed 4 about 8-week-old calves. We fed them infected milk for 4 days and then began feeding them uninfected milk. We necropsied 2 of the calves at day 6 and 2 of the calves at day 13, just to give us an idea of what acute infection looks like, but then also enough time to say, okay, are they making antibodies? So we fed calves this infected milk from our experimentally infected cows. And here are just kind of the results of our clinical observations. So here we're on a 0 to 3 score. 0 would be completely normal, healthy calves. 3 would be calves with really lethargic behavior, really unusual feces, a lot of nasal discharge, or like really acute respiratory distress. And you can see right here, we didn't really get anything above a score 2. So we did see some mild lethargy in the calves, some nasal discharge, some increased respiration. But if anybody has raised calves, that's incredibly common. It's a lot like having a child in daycare. They've always got something going on. And we did see just a little bit of mild fever in these calves. So then the question is also, okay, if this is infecting calves, where is the virus going? And what is the immune system doing? So we took a lot of different samples to see is there a reliable test for this. So we did not see any virus in fecal samples or blood samples from these animals, which is pretty interesting. We did see the occasional ocular swab or the occasional saliva test pop positive, but we definitely saw consistent detection in nasal swabs from these calves. And beyond just when they were eating the infected milk, so once they were in clean milk, they were still testing positive for nasal swabs, so we knew it wasn't just cross-contamination. This was actually an infection of these calves. And then we further confirmed this infection because we were able to detect antibodies in these calves as well. When we necropsied these calves, we saw some signs of respiratory disease. So we saw here what you can see in like the little black arrowheads are some small lung lesions. And then the larger black arrows point to actually enlarged lymph nodes. So we are seeing kind of more of a classical respiratory infection from influenza from these calves consuming this infected milk. To share, this is kind of one of those lung samples. under a microscope that's been stained. So you can see kind of outlined in teal is where our actual lung lesion is. So you can see where that tissue is super consolidated and closed, as opposed to the open, healthy airways that surround this lung lesion. And within this lesion, we are able to see virus. So you see kind of that zoomed in picture with the green arrowhead is stain confirmed virus, so that we can say, yes, these lung lesions are being caused by this influenza virus contracted from consuming this raw milk. So this was nice because we had a larger number of calves. We were able to look at gene expression. And so what that does is lets us look, instead of just trying to focus on one aspect of the immune system, to understand overall what the entire immune system is doing. We tend to think of genes as something that are inherited and are just kind of stagnant. That's really not the case. All the time, genes are turned on and off to make proteins. So what we're able to do is look at that intermediate. So genes are transcribed into RNA, which is translated into protein. And so if we're able to sequence this RNA, this intermediate step, we can see what genes are turned on or off during certain conditions. And so that's exactly what we did. We took daily blood samples from these calves, both before they were infected and then every day during infection, to say, okay, compared to pre-infection, what genes are turned on or off in these calves. So that gives us a really nice overview of what's going on in the immune system. So here we have a plot. And so this is just day 2, because that's kind of when we saw the strongest infection, to say, okay, what genes do we see being turned on or being turned off? So we have genes that are turned on or upregulated, shown in red, versus there's a pretty small number in comparison that were turned off in green. And what really jumped out was this gene CBP1, or Z DNA binding protein 1. We saw this gene turned on at every day of infection compared to the calves pre-infection. So what does this gene do, and why was it so exciting to us? It recognizes and binds zRNA structures that are produced by influenza virus infected cells. And so of course you were like, okay, yeah, this influenza virus infection is going to turn on this gene that's really important to influenza virus infections. That was a really exciting finding, because let's go back to, we did not realize that cows could get influenza A before this. So knowing that they are having a very similar immune response to other species that have been known to be susceptible to influenza A was actually a pretty exciting finding. This means we don't have to start from scratch. We can learn, or we can use a lot of what's already been learned in other species to kind of move forward and inform studies, inform vaccines, inform interventions. Because this gene here is really kind of what's known when the body says, hey, we are recognizing this influenza virus, and then it basically starts the immune system and says, okay, we need to respond to this virus. We also looked at, how do these genes respond within the pathogenesis of influenza pathway? Because the immune system can kind of be tricky. We like to talk about what's called the Goldilocks theory. So you don't want to have too little immune response where the virus just runs rampant and causes infection. But you also don't want to have too much immune response where you're getting some inflammation and some other harmful things, because it kind of just goes too far the other way. So it was really nice. I'm not going to try to show you guys this entire pathway, unless you want to take a nap this afternoon, because it is lunchtime. I'm just going to zoom in on what was kind of the really cool finding of, okay, this is what genes are going on post-infection, and kind of what's known. So just kind of a quick overview of what's going on with the colors here is anything that's orange means that's a pathway that's being activated. And anything that's in blue means that's a pathway that's being inhibited. So it's very exciting to see right away what is being inhibited: viral infection. So these calves are posting enough in an immune response that they are going to inhibit viral replication. They're going to activate antiviral response. But what's really nice is that we are not seeing these immune responses go to a point where we're going to see severe lung pathogenesis or hear acute respiratory disease syndrome. And so these are things that you would see in super susceptible species like poultry, where actually the immune system kicks into overdrive and starts to cause some of these lung problems. And so that's really good news, is that we are seeing kind of this sweet spot where we have enough immune response to inhibit viral infection, but not too much to the point where we're causing any kind of other pathogenesis. So to just kind of summarize, unpasteurized milk can infect calves. So waste milk should be pasteurized before being fed to calves. Infection induces a known influenza A innate immune response, but not to the point where it causes like an acute respiratory distress syndrome. So really good news on the calf side as far as what we're seeing for an immune response. So kind of what's next is going to be vaccine studies. So the nice part about what we've done is we have now an experimental model. So we know in lactating cows that we can recreate the disease in experimental conditions. And so that gives us the ability to test vaccines. So you can have vaccinated and unvaccinated animals next to each other, exposed to the same virus in the same way. And that's going to be able to effectively tell us, does this vaccine protect against disease? It's also nice because we don't have to do everything in lactating cows. Obviously, that's the gold standard if those are the animals we want to vaccinate. That's obviously not the only time that animals are vaccinated during production systems. So we also have this chance to test this vaccine in our bucket calf model. So that gives us a way to understand how young stock can be protected and a way to test a larger number of animals without having to go and put into lactating cows in a BSL-3 model, because we've talked about that just takes a whole lot. And so we are going forward with DIVA vaccine studies. This is another term that I want to talk about. So unlike what my husband thinks, DIVA does not just mean I worked on it. This is an acronym that says differentiating infected from vaccinating animals. So as we move into surveillance for this virus via bulk milk testing, it's really important, as we also work to protect animals with vaccines, that we are able to differentiate infected from vaccinated animals. So that's what we're pursuing in both young stock and lactating cows, is how effective are these DIVA vaccines as far as protecting animals. And then also what's next? So we have started bulk milk surveillance testing. Previously, we thought the incursion of highly pathogenic avian influenza into cows was kind of a one-time thing. We really thought this was a single spillover event into birds. Bulk milk surveillance testing, unfortunately, has told us that is not the case. In the last 2 weeks, we've had confirmed cases of a new genotype. So hopefully everybody was paying attention in the beginning, and you're better than some of my former students. And you recognize that, hey, she was talking about this genotype B313. In the last 2 weeks, we have now identified a new strain, D11, in cattle in Nevada and Arizona. And so this strain, D11, has been the one that's been circulating in wild birds for a long time. This is the first that we have detected it in cattle. It is not like the previous strain tied to any kind of movement of cattle at this point. It seems to be multiple introductions from birds into cows. So this now means we have a new set of tasks on our plate. So we are looking into, what does the actual pathogenesis of disease from this genotype look like? Because initial reports indicate that it's not as severe as the first. We're seeking to try that under experimental conditions. And then 2, we've just shown that these antibodies to B313 protected cows against reinfection. Are cows that have had B313 going to be protected against D11? So we've identified some cattle with antibodies. We're working to test these, but that's kind of what is next for studies here. So with that, I know I've thrown a lot of information about flu and the immune system at you. I'm super happy to take any questions, and I would really like feedback if we have producers on here as kind of what are the priorities for producers, given that we have a limited ability, what are the most important questions for us to answer first? And so with that, I will take any questions.
Okay, I've got a couple of my colleagues who will be helping with questions. Kaitlyn, you can take a look at the chat box as well. Jan, why don't you start us out with a question from the audience?
Sure. I'll just use the first one that came up here, and it's asking, when you're talking about scarring or fibrous tissue in the mammary gland, is that damage a permanent condition, like scar tissue in your knee, for example?
That's a great question, and one I can relate to with the scar damage in your knee. Yes. And so that's the unfortunate news, is that that kind of scarring in the mammary gland is unlikely to resolve. And so we would expect the next lactation for infected animals might be impacted. And I think we're about to the point where we're going to see that. I think a lot of these, or some of these previously infected cows, are kind of ready to go into their next lactation. I think we're going to see some of that.
And it looks like the next question here, on your viral detection slide, you mentioned you considered CT less than 35 positive. Do you have a virus isolation data that supports this is a good cutoff value for positive cases?
That is a great question. And yes, we do. We are typically able to isolate a virus if we see a CT less than 35. What is interesting, we did viral isolation, especially from the milk samples I talked about in the first study. But once we got to about the day 14 point, where we had both this virus at the levels below a CT35 and antibody in the milk, we were no longer able to isolate virus. And so that leads us to believe that actually the antibodies were binding up the virus in the milk.
When you were talking and you indicated you infected 2 quarters,
Mhm.
was it just 2 quarters? We have a question. Did you inoculate all 4 quarters?
No, so in this, we only inoculated 2 of the 4 quarters, and I apologize for not making that more clear. And so it was pretty interesting because we mostly saw the infection stay in those 2 quarters and not cross over to the 2 uninfected quarters.
And then kind of going back to the calf side of things, do you plan to do a study to reinfect calves to see if they will hold the immunity?
Yes. So the first subsequent calf study that we did was to then, so yes, that's a plan. We also wanted to see kind of how we were talking about the virus isolation wasn't possible from the milk once the milk had antibodies. We actually took that milk that had both virus and antibodies and fed it to calves. And we are working through all of the data currently, so I wasn't quite ready to present it, but preliminarily it does look like that protected calves. But it doesn't look like they were able to acquire any antibodies from that. So we were kind of hoping it would go that way, right? If we fed them this milk with virus and antibodies, that they would be protective, but also maybe build their own antibodies. They didn't. So we will go back to feeding them the infected milk. They make antibodies, and then we see if they're immune.
We
That might be-
saw, go ahead, Jen.
Oh, I was gonna say it might be a follow-up question to that is, how long does that natural immunity last? And maybe you kind of answered that with your response
That's a
there.
great question. I know ISU's got a lot of work they're doing to follow naturally infected cows and see how long antibodies are lasting in them. Because it's hard to maintain cattle in this BSL-3 environment for a really long time. So I think that's a better study question for the field.
We saw in the popular media an article citing some UC Davis research on milk acidification instead of pasteurization for raw milk being fed to calves. Anything you looked at able to relate to that?
So I should probably make that clear that pasteurization does inactivate this virus. That's very well known. The big push to look at that acidification is a lot of waste milk going into lagoons and then that being spread on farm fields, etc. I have not had a chance yet to see if acidification would inactivate the virus fed to calves. That would be a really interesting follow-up question. Thank you for that.
There's a question here about the different strains. So is the D1.1 affecting animals the same as the B313?
At this point, it's a little soon to tell. Initial field reports are not reporting as much disease as far as drop in milk production or rumination or discolored milk that we saw with the initial B313. So that's our goal in the next few weeks is to do an experimental infection with D11 and compare it to what we have already seen in B313. And are we going to see the same thing?
I would encourage folks to type in questions. We're still a fairly large group, so we really need to do it this way. You had mentioned with the DIVA ID infection versus vaccination. This gets into a marketing question. Would that be able to be a way to say, we don't have the infection, we have vaccinated the cows that produce this milk?
Yes, absolutely. So that's a lot of what is so important about that DIVA vaccine protocol.
How broad a spectrum do you think these vaccines will be? Can we do the B series and the D series and whatever the next series is with a singular vaccine?
That is the million-dollar question. So the thing that makes flu so difficult is it is so picky about antibodies. And so I think our upcoming challenge of cattle that have the B313 antibodies, and if they are susceptible to D11, is going to inform a lot of that. So I obviously really hope so. But I think it is too soon to tell. And I will say my influenza colleagues are not super optimistic.
And then we had a follow-up question regarding the fibrous or the scarring tissues, because theoretically some of those cows that were infected early on might be in their subsequent lactation. So are we seeing any effects on somatic cell count or decreased milk production from that?
That is a great question. As far as I know, we do not quite have any data to answer that yet. But again, that's a really great question for the field. So somebody may have data on that I'm not aware of.
Have older animals been challenged orally with raw milk infected?
Yes. So have older animals been challenged orally with raw infected milk or other virus positive materials? The answer is kind of. So in the same publication that I was talking about, a lot of our lactating cow work, we also challenged some yearling heifers with kind of a classic respiratory disease model. So we actually use nebulizers that are typically used to treat horses and fill those with virus. And so we did see that infect those yearling heifers, but with very, very mild disease. So less lung lesions and things than we saw in these these bucket calves. And so some of that could be due to the age of the animal, or some of that may be due to, it seems like milk is just a substrate that this virus loves to live in. So it's kind of impossible to tell those apart at this time. If you have suggestions on how to get some yearlings to drink milk out of a bucket, I'm all ears. Because I've been asked if I could do that, and I don't know how to make that possible.
Danielle asked the question, are there plans to expose naive lactating cows and calves to the D1 series?
Yes, so that is the upcoming plan. We are going to expose naive lactating cows to D11 and see what pathogenesis looks like. How does that compare to B313? At the same time, we will run them along cattle that have the B313 antibodies to answer both questions. Is what does this disease look like in naive cattle? And do these B313 antibodies protect against D11? And I appreciate the used jerseys. That would, yes, that would be one model.
It looks like there's a comment in here from Dr. Danelle Beckett-Whittle about UC Davis doing some work on acidification in the field for calves being fed waste milk. So that lab study benchtop is promising. So taking that to scale is appreciated.
That would be very cool to see.
We have a question. How does the virus transmit from cow to cow and herd to herd? Do you want to kind of share some information on that?
Absolutely. Bad news is we don't have all the answers. So we were definitely able to trace the movement of this from herd to herd with infected cows. But within the herd, how it's moving from cow to cow is not something we've really been able to nail down yet. And a lot of that is just the limitations of how many animals we can run in BSL-3. So if this is something that happens, 1 in 10 times, and we're only able to run this study in 6 cows, we just don't know, are we not seeing it because it doesn't happen or because we didn't try it enough times? So it's been really hard for us to nail down the transmission question. It's also really hard to nail down the transmission question in BSL-3 conditions. So it's very clean, very sterile. For example, we have constant circulating air with lots and lots of HEPA filters. The air changes over every 6 minutes. So the virus is also maybe not able to hang around in these experimental conditions in the way that it would on the farm. And so we've got a lot of folks working on the epidemiological side, and I'm hoping we can get some more answers from that.
Okay, we've got,
Um,
go ahead.
I was gonna read Elizabeth Cox's question. So it sounds like feeding infected milk is a way to vaccinate calves to develop their own immune protection. I would strongly discourage against that because of the nature of influenza viruses. If these calves are becoming infected, you are giving this virus a chance to mutate and a chance to reassort and become better able to infect calves. And so we don't want to do that. So I would discourage calf infection. I know from when calf infection is so mild, it's really tempting, but we don't want to get this out there in a way that it becomes more severe for these calves.
Christopher asked if you were able to sequence viruses several days post-infection to look for PB2.
Yes, and so if you are not familiar with PB2 mutations, that are mutations that make this virus better able to infect mammals, that work is ongoing. So we do not have any results on that I can share yet.
And we appreciate Marissa put in there in the chat the link to an article on milk acidification. So the first of few intended for that. So if people want to click on that, they'll be able to get to that article.
If you're going to identify a singular thing that you need to be looking at in the next battery of research, what is that?
I would love it if you could answer that. So the two questions, right, that seem to have come up as the most pressing questions are, what does the D11 look like in naive cattle, and what does it look like in cattle with B313 antibodies? So those are the two questions we are set up to answer in the coming months. Hopefully it doesn't get any more widespread after that. So I would love to hear, what are the pressing needs for dairy producers? What are the questions that y'all would prefer to see answered?
I've been looking to see in our list if we've got any poultry producers that I can identify. You mentioned that the D11 is the predominant clad in poultry. So are we looking for transference, or what are we looking at really between the two species?
That's a very broad question. So with B313, we did see that strain move from cattle to infect poultry. So that's obviously something we want to prevent from happening, because this is highly pathogenic, even influenza. This is incredibly devastating to poultry producers. With the D11, I think the question is kind of, is it going to go the same way? Is this going to become widespread in dairy? Could that be a route to reinfect poultry? Kind of what can we do to stop the spread? And then it's kind of a question of, we really thought this was a single spillover event into cattle. It's incredibly surprising to see it again. And so the greater question of, is this something we need to be prepared for going forward? Kind of, what do these other strains in cattle look like?
There is a question in here, and I think we've gotten this a lot, is any of this research have the potential for infecting beef cattle, or any research on beef cattle?
That is a really great question. Based on what we've seen, I think it would be highly likely for beef cattle to become infected. I think seeing that calves can become infected from consuming infected milk means that this could spread from cow to calf and cow-calf operations. Then, of course, if we are seeing this mild disease in calves, and these calves would be bull calves or beef on dairy calves that would be going into a beef system, I think you have the ability for it to move into beef there also. And this could potentially become part of the respiratory disease complex that plagues feedlot systems. It's been discussed, is it worth challenging beef cattle? I think it kind of comes down to, do we have a good model that works in non-lactating animals? And then kind of, where would we want to go with that for beef? Like, do we have a really good way to surveil non-lactating animals, et cetera? So it could go into beef. I think we need some direction as to what are the important questions. The other thing I would say, because we've seen this huge outbreak in California, we've seen quite a few abortions of infected cows. I think that would be the other place where this could be economically really challenging for beef producers if you have bred cows aborting. And I think that might be the best way to look at this, is I think that's a question that affects both types of producers. If we have these pregnant cows aborting, I think that's a challenge for both dairy and beef producers. And I think that might be the way to go looking at that. But again, it's a constant struggle to, okay, we can answer one of these questions at a time. Which question do we ask first, right? So, do I think that's a really interesting question? Yes. But if we're not having a strong economic impact to beef producers yet, we might need to focus our limited resources on who's being hit hardest.
Our colleague from Michigan asked, you said that in experimentally infected calves, the virus was found in nasal swabs, but is that true in adult cows as well?
That depends on the route of infection for adult cows. So we did not see that in our experimentally intramammary infected cows where we saw it go and cause positive nasal swabs, but we have seen positive nasal swabs in the field.
Okay, Jan, are you seeing additional questions coming in, or Philip? We haven't heard from you. Do you have a question?
No, these are very good questions on here. Let's thank Kaitlyn for answering them so well.
Thank you.
There was a question about, is this recording going to be available? And so it will be on our I-29 Moo University website. And we also have a YouTube channel. So once it gets edited, it'll be out within the week.
Jacob asked, has anyone done any serology surveys in neighboring beef herds?
Not that I'm aware of. That's an interesting question. I don't, Phil, do you know of anybody? You're more in touch with the field work, I feel like.
I don't know of anybody that's done that. We get asked that a lot when we give talks on the high path, whether it affects beef herds. And the answer seems to be no, it doesn't seem to affect the beef herds very much, but we're not really looking that hard either. So is it because we don't look, or is it because it really doesn't affect them? We don't really know. But when you think of how they're managed, they are managed quite a bit differently, right? They don't come through a parlor. Usually just one animal is nursing that cow at a time. So they are different animals, but they still have an udder. So it makes sense that they might theoretically at least be susceptible.
We've got a few minutes left in our time. So if folks have additional questions, now's the time to type them into the chat box.
I asked Kaitlyn to talk about the CITO program that went into effect in December. I know that there were a few states that had not been started yet, and I heard today that they're going to be tested. Kaitlyn, do you know any update on that?
I don't. So from within USDA, we have separate branches. So I work for the Ag Research Service. So we perform research. The testing is under the Ag Animal Plant and Health Inspection Service, so APHIS. So they are rolling out the testing. Last I heard, they were pretty confident that they were able to cover most lactating cows, but I have not heard any more recent updates.
I do know that Wisconsin and Idaho and Illinois had not been added on the map, but they're on there now as participating. So looks like it's going to be almost the whole herd, almost the whole 48 contiguous states are going to be participating now.
Philip, go ahead, excuse me.
No, go ahead.
You had a really good comment and fits into Philip Durst, Michigan's question. What have been the results of the USDA bulk tank testing program? And you commented about the Nevada and Arizona.
Correct.
Yeah, so that is how we found the herds in Nevada and Arizona that had the D11. So I think that's a very positive thing. It shows that it's working and helping us find this new strain before it got much further. And if we hadn't been doing that, we wouldn't even know it existed, maybe. And then it looks like most of the rest of the country is truly negative right now. And I think to be officially called that, it's going to take several months of continual negative tests. But so far, there's 3 states that are negative: Pennsylvania, Mississippi, and I can't remember the third one, but it's that were testing before the mandate came in December. But yeah.
Jamie Yonker has noted that we've got 3 states that are the final states to approve surveillance plans. Jamie, if you want to unmute, you can maybe kind of follow up on that.
Yeah, and I can introduce myself real quick. My name is Dr. Alex Turner. I'm the National Incident Coordinator for the High Path AI Response in Livestock for USDA APHIS. Our national milk testing strategy is down to the last 3 states of North Dakota, Florida, and Massachusetts that are yet to have approved surveillance plans, but we have the other 45 states representing 99.4% of the herds that contribute to grade A milk in the US are currently either have plans or are actively testing and looking. So the states are at different places along their testing pathway, but you're exactly right. The Nevada case and the Arizona case are both would not have been detected. And speaking from personal knowledge at the Nevada cases, talking to the state animal health officials out there, I think they're up to about 9 dairies in that same valley in Churchill County that have that same strain of D1.1. Only one of those 9 had any clinical signs that would have been picked up by any veterinarian or any dairy person. The other 8 have not had any. The Arizona case and the neighboring dairies have actually had an increase in their milk production through this whole time before and after that's been detected. So I would say that that also equates to 0 clinical signs in how the D11 is behaving relative to the B313. That obviously is a very small sample size, but I can say very confidently without the national milk testing strategy, we would not have found this at all. D1.1 is, it passed last week the #1 genotype for all poultry detections coast to coast. It is in all 4 flyways of wild waterfowl and migratory birds from the Pacific, the Central Mississippi and Atlantic flyways all have this version of the virus, and it is impacting poultry production in all parts of the country, north to south, east to west. So these type of additional spillover events, certainly the hope is that it does not do what B313 did and get spread throughout everything else, and in stopping the movement of those cattle in Nevada at an early stage, because the one place that was had any clinical signs, the test was positive prior to any of the physical clinical signs in the cattle that had it. Still more mild than the B313 seems to be across what we're hearing from out in the field, now with 975 infected herds. So yeah, just a quick update from our side of the shop. I enjoyed the presentation and thought I'd just chime in with that since some folks were asking questions.
Thank you.
Yeah, thank you.
Can you give us best guess? What's the link between those 9 Nevada herds? I mean, is there a commonality there?
They're currently looking at that. We do have tactical epidemiologists on the ground looking at that right now. The genomic testing won't be, because of the time frame and the difference there, no amount of analyzing the genome there will be able to differentiate whether it was a single spillover event and 9 lateral movements or 9 identical spillover events. It sure is less likely, but was it a single species of migratory bird that brought it there? That valley in Nevada also has a mutation in the mammalian PB2 gene of the D701N. And that's one of kind of the 4 mutations they keep an eye on. And that mutation, it suggests that it might have actually gone through from a bird to a mammalian host to the dairy in that order, but again, we can't necessarily confirm that. And unless somebody comes up with a smoking gun of like, hey, this raccoon died in my waterer, or this cat fell into the milk, bulk tank, we can't necessarily say that's what caused it. And that still may be the case, right? They've had, we know cats are very susceptible to HPAI, not just a specific strain, all strains of HPAI, of H5N1. Cats get sick with this, and it is a dose-dependent sickness, and a lot of them die. So that's one animal, at least, that we know doesn't do particularly well, but also as a peri-domestic animal, there's plenty of feral cats around dairies because guess what? There's plenty of rodents around dairies, plenty of food around dairies. So we don't have enough from the field to supplement to answer all those questions. If anything, certainly we are looking at that valley in Nevada for commonalities, potential for spread. It is a very small defined area. It's an area that's kind of a very tight-knit community, like a lot of the dairy communities you all live and work in. And it's geographically limited because it is kind of so far in a single valley in that county.
Thank you. Alex or Kaitlyn, Danielle brought up an interesting circumstance because of Massachusetts there. Do you want to take a look at that?
Yeah, it's not through a NALM lab, which is the National Animal Health Lab Network, and it's not through the NVSL. They're using the Broad Institute lab since last summer, and so the state of Massachusetts feels comfortable with those results. The national milk testing strategy kind of lined out the acceptable regulatory results. The Broad Institute, no matter, I'm not personally familiar with that lab as a lab system. It's not in the regulatory lab chain, and so as such, those that doesn't mean they're doing bad work there. I don't want to say that there's nothing there, right? We know Massachusetts is looking, but Massachusetts wants to continue to look with that lab as their testing lab. That lab is not a NALM lab. It's not a part of the National Animal Health Lab Network. It's not a diagnostic lab. It's a research lab, which is slightly different in terms of how samples are handled and everything else. I'm not trying to question that. They also have not had any detections testing through that lab. So we're still trying to work with the state of Massachusetts and the milk regulators, and we may be able to still work something out to get them to participate and have regulatory stuff. Yeah, and Jamie, thanks for pointing that out. I'm again, not questioning the quality necessarily, just saying it is not a diagnostic lab. It is not a regulatory lab.
Maria asks, can we find the counts of the genotypes within the USDA website?
I believe it's not a USDA website, but I think the National Veterinary Services Lab has been sharing those genotypes on through a known process, and I always forget the acronym for that process as not a lab person myself. But those genotypes are being shared publicly, I believe. For the actual genotypes on infected prems, on infected dairies, those go up after a certain period of time passes for public consumption, and that's all in the same place. And I apologize, the acronym escapes me. It's not on the tip of my tongue. Yeah, John Corsland, it is a common virus. It's all the same kind of branch of genotype there in Nevada. GISAID, yes, thank you, Steve.
Okay, if we've got any other questions, we'll just wait a second or two while we're waiting. Dr. Kaitlyn, any final comments from you?
I don't. Thank you so much for the opportunity to speak today.
Very good. I'm not seeing any additional questions coming in. Appreciate the time you folks have spent with us today. We had Dr. Kaitlyn gave us a lot of great information and a lot of neat input from colleagues around the country. We appreciate it. Because you registered, everyone who registered will be receiving notification when we have this in the archive. So give us a week or so and we should have it available to be used out there in the countryside, so to speak. Thank you, Kaitlyn. Appreciate the time, and we'll look forward to having folks back at our next monthly webinar.